Alright, this is a review of the nervous system. We are going to just kind of do a quick rundown of all the topics we've discussed so far in this nervous system unit. And I did post a one-page, a little informational sheet, study guide if you want to call it, to help organize your thoughts as you approach this exam coming up next week. So looking at our notes here, we talked about the sensory information comes into the brain where it's processed, and then we have a motor response. We organize the nervous system of central nervous system, peripheral nervous system, motor, sensory. Remember, the blue arrows represent information coming in, red arrows information going out, and then we took that motor and information going out and characterized it into somatic, which serves skeletal muscle, voluntary control. Autonomic serves smooth muscle and cardiac muscle and glands under involuntary control, and then we finished up talking about the parasympathetic and sympathetic autonomic responses and their effects on the organs. So we looked at the type, the typical neuron, the multipolar neuron, the dendrites, cell body, axon. The axon is heavily myelinated. Remember, it has voltage-gated channels at these nodes of RAN-VA. That's where the action potential is generated, or I should say propagated, where it travels along the axon. The actual location where we achieve an action potential, where it begins, is here at the axon hillock. So out here in the dendrites is where we have graded potentials developing. It's unmyelinated, and those can be IPSPs or EPSPs, depending if it's depolarizing the membrane, then it's an EPSP, it excites the cell. If it hyperpolarizes the membrane, it's an IPSP and will inhibit the cell from achieving an action potential. And then remember, down here at the axon terminals is where we see the release of the different neurotransmitters. Two major ones we talked about were acetylcholine and norepinephrine, depending if it's a sympathetic neuron or a parasympathetic neuron. So if you look at channels, remember we have chemically gated channels out here on the dendrites. We also might have a special neuron that has sensory receptors on the receiving end of the cell. So to get an action potential, we have to have enough of a stimulus via those sensory receptors to open up channels to special channels that are sensory receptor specific, like photoreceptors, mechanoreceptors, nociceptors for pain, proprioceptors if we're talking about the muscle spindle or Golgi tendon organ. All those are examples of special channels that are specific to a sense. Photoreceptors for light, thermoreceptors for temperature, and so on. So we have chemically gated or sensory receptors, specialized channels on the dendrites, and then voltage gated channels along the axon, and also on the axon terminal we have those voltage gated calcium channels. So we talked about different types of channels. This is a chemically gated or ligand gated channel, so here it requires something to bind in order to open up the channel. And in this case, it looks like sodium is passing through into the cell. It shows potassium coming out. That's not typical. Normally we see one type of ion moving through a channel. So either way, just know that a chemically gated channel requires some chemical to bind to the channel in order for it to open. So we find chemically gated channels again at the dendrites and cell bodies. Gated channels we talked about require some difference in ion concentration on either side of the cell. We call these voltage gated channels because they also can be stimulated to open if we simply run an electrical impulse through the membrane. But in our cases that we studied this in this unit is we're looking at ion concentrations, and if it becomes different on either side of the cell, we say it's polarized, and if it becomes the same, then we say it is depolarized. Locke talked about resting membrane potential. We have more leak channels for potassium than we do sodium. We also have the sodium-potassium pump, and we have negatively charged proteins inside the cell, all of which contribute to resting membrane potential. And the resting membrane potential is negative 70 millivolts in a nerve cell. So anytime we cause positive ions to flow into the cell, or the inside of the cell becomes more positive, we call that a depolarization. If it becomes more negative, any flow of an ion that would make it more negative inside the cell, we call that a hyperpolarization. Graded potentials are special potentials that cause the change in ion flow on either side of a membrane that travels in two different directions and they fizzle out. So we need a stronger stimulus if we want to generate an action potential that only flows in one direction. So graded potentials can travel in two different directions. Action potentials travel in one direction down the axon. Again, remember these graded potentials are occurring out here at the dendrites and cell body. So here we talked about an action potential. This we call resting membrane. -55 we call threshold. And we have depolarization, repolarization, hyperpolarization, and then the sodium-potassium pump bringing things back to normal again. There's not going to be a lot of questions on the action potential, only because we studied that in general A&P. So I'm more specifically going to ask you about what type of changes in the membrane would cause hyperpolarization or depolarization. So I'm looking more at your understanding of concentrations of ions on either side of the membrane. Remember that chloride, sodium, and calcium are found in higher concentration outside the cell. So if I open up a channel on the membrane, they want to flow in. And potassium and magnesium and intracellular proteins are found in higher concentration inside the cell. So if I open up a channel or allow movement of a protein, they're going to flow out of the cell by their concentration gradients. So here this slide talks about stimulus strength and how that affects action potentials. The stronger the stimulus, which is shown down here, the more action potentials we're going to get. So if we're looking at a moderate level sound versus a very loud sound, it's just going to generate more action potentials the stronger the stimulus. So we can't get a higher magnitude action potential. We can only increase their frequency when we increase the stimulus strength. So the absolute and relative refractory periods, if I apply a stronger stimulus here, I can get another action potential to begin right after this one is done. But I cannot generate another action potential in the middle of one that's still going because of that absolute refractory period. Talked about myelinated fibers. Remember, the more myelinated it is, the faster the impulse will travel down the axon. So heavily myelinated axons are what we see with skeletal muscle. So we see rapid transmission. Different types of connections between neurons for transmitting signals. We talked about the chemical synapse. Remember, we have acetylcholine or norepinephrine in these vesicles here. In this case, if it's going from one neuron to another neuron, that's acetylcholine. And these are chemically gated channels on the postsynaptic membrane that will open and allow sodium in. Remember, calcium is flowing in here as an action potential comes down, causes acetylcholine to release its contents into the synaptic cleft. So this is how we can get an action potential in one neuron to be generated in the next neuron. We talked about postsynaptic potentials. This is an EPSP, this is an IPSP. Both of these are graded potentials. They occur at the dendrites of the postsynaptic neuron. So these potentials add up to either reach threshold or not reach threshold. Here the two added up to reach threshold and we got an action potential from them. Here we had two EPSPs, one after another, not strong enough to reach threshold, so we did not get an action potential. Then we talked about the spinal cord. Definitely know the anatomy of the spinal cord. I'm not going to ask you specific questions on anatomy, but I'm going to ask you functions or what type of fibers we find in the different parts of spinal cord anatomy. So we know that sensory neurons travel through the dorsal root. We know that the cell bodies of sensory neurons are in the dorsal root ganglion. We know that the sensory neurons synapse on the dorsal horn of the gray matter, and then they travel up the the sensory ascending tracts to the somatosensory cortex or the cerebellum. We know that the ventral root carries motor neurons away from the spinal cord to the effector organ. We know that the cell bodies of those motor neurons originate in the ventral horn of the spinal cord. Remember, this is the lower motor neuron. Starts in the ventral horn and then goes out to the skeletal muscle cell. We know that the white matter of the spinal cord is made up of myelinated axons, and those axons are clustered into different tracts. So those tracks can be ascending or descending, carrying sensory or motor information. So here's just some of that information that comes in. Again, these are all going to be sensory neurons. These are all motor neurons, somatic or autonomic. Talked about the reflex arc. Talked about the different motor tracks. And when we look at the motor tracks, I went over this nice chart for you. And I've gone through and kind of highlighted some of the unique information for each of the tracks. So be sure that you know the names of the tracks and what sensation they carry. And the first, second, and third order neuron, a lot of these are the same information. Just kind of know where they start, where they synapse, and where they end up at the end. All important information. The motor pathways, we talked about 3 of those also. Just know basically where they cross over, where the lower motor neuron begins, where the upper motor neuron begins. Important information there too. So we talked about the different tracks. You can look at them here to kind of have a visual for those tracks. And then we talked about the peripheral nervous system, the segments of the spinal cord, how we have different levels of control of sensory information. Remember that the muscle spindle and the Golgi tendon organ, those are the 2 proprioceptors that deal with body position. So Golgi tendon and the muscle spindle. The Golgi tendon helps measure and control muscle tension, and the muscle spindle helps control and sense, or it contributes to the control by creating sensation of the stretch or length of the muscle cells. And then we have sensory receptors in the skin. Pain, touch, temperature are examples of that. So remember, we have the first order neuron, the second order neuron, and the third order neuron. So again, know where those neurons begin and end. Talked about the different levels of the spinal cord. And then we talked about levels of control. Remember, we always have reflex activity happening that's beyond our conscious awareness. We have the tracks that we do have under control, motor tracks, and then we have those higher centers that we are not aware of, and that's the basal nuclei and the cerebellum that help control information going out. Again, the reflex arc, the muscle spindle. We have the alpha motor neuron that controls the contraction of the quadriceps muscle to keep us standing upright, contributing to muscle tone. And then we have the sensory receptors wrapped around the intrafusal fibers, which if they become slack or are not stretched, then that stimulates the alpha motor neuron to contract and keep us with proper muscle tone. We talked about the reflex arc and how it controls muscle tone via the muscle spindle. So we went through each of those steps. This is a very important diagram. I would have this ready for the test. Be sure to label the different parts of the reflex arc for the patellar reflex. Definitely going to be on the test. Then we talked about the autonomic nervous system. Lots and lots of really good information here. Definitely look at this diagram, know the details of it, label what types of neurons we have here. These are all cholinergic neurons. This is a cholinergic neuron. This is an adrenergic neuron. And the receptors that we find here, this is a nicotinic receptor on muscle cells, nicotinic receptors here, nicotinic receptors here, nicotinic receptors here. And then we have the adrenergic receptors here. I'm sorry, the muscarinic receptors here. All the parasympathetic organs have muscarinic receptors. And then with the sympathetic nervous system, we have the alpha, beta 1, and beta 2, and so on receptors here responding to norepinephrine. So we compare the layout of the sympathetic and parasympathetic. Remember, this is the craniosacral division. This is the thoracolumbar division based on where those neurons come from the spinal cord. We talked about all these, again, all these receptors are muscarinic receptors, and these are all cholinergic neurons in the parasympathetic nervous system. Sympathetic, these are all cholinergic neurons. These are all light green. These are the adrenergic neurons, and there's different receptors depending on the organ. Some are beta 1, like in the heart. Some are beta 2, like in the lungs and most of the other organs, and some are alpha, as in the skin and the blood vessels. So this is just a table that summarizes a lot of the information. I think it's good to make it visual by applying the information to the pictures of the parasympathetic and sympathetic nervous systems. And then do know the general effects of the sympathetic and parasympathetic nervous systems on these major organs. And recall that the 3 organs that do not have parasympathetic neurons are the sweat glands, adrenal medulla, and the erector pili muscles, and also, I should say again, the blood vessels, the coronary blood vessels serving the heart. They do not have parasympathetic neurons. In all the other cases, all those organs have parasympathetic neurons and sympathetic neurons. And we talked about referred pain is where you can have a gallbladder that's inflamed, which sends information through those visceral sensory neurons that enter the spinal cord and share the same pathway as information coming from somatic sensory neurons, and we feel pain in the shoulder related to pain in the gallbladder. So they share the same pathway to the spinal cord, so we can have a sense of what type of internal issue someone has based on the pain they describe. So looking at the study guide that I prepared for you, these are the diagrams that are very important that you should label and know the details of. And then be able to answer these questions. I think I went through most of them. I guess this one, tract versus nerve. A tract is a bundle of myelinated axons inside the central nervous system. A nerve is a bundle of myelinated axons outside the central nervous system. A horn is bundles of cell bodies and dendrites inside the spinal cord. Ganglion, bundle of cell bodies and dendrites outside the central nervous system. And the rest of these we talked about in class or in the video, so be sure that you are able to answer these questions. If you have any confusion, you can watch the videos again or refer to your textbook. If you have any questions other than that, you can see one of the tutors or shoot me an email.